esta ceremonia inaugural del SOMI 34 Congreso de Instrumentación. Les ofrecemos una disculpa, el retraso del autobús, son cosas imponderables que pues no está en nuestras manos, pero lo importante es que ya estamos reunidos aquí y quería yo presentarles a quienes nos acompañan hoy en el presidio. Primero es el doctor Rodolfo Zanella Especia, el director del ICAT, que nos ha hecho el honor de venir a inaugurar el evento. Rodolfo, muchas gracias. ¿Sí? El doctor Joel Vargas Ortega, que es el jefe de la unidad Morelia, de aquí del Instituto de Investigaciones en Materiales, aquí en Morelia, y que pues son en esta ocasión nuestros anfitriones, cosa que agradecemos y reconocemos mucho, doctor Joel. Y un servidor, Gerardo Ruiz, en representación del comité organizador de este congreso, a todo el comité organizador, tanto local como el de Ciudad de México, pues un reconocimiento y un agradecimiento enorme por su trabajo. ¿Sí? El doctor Joel Vargas nos va a dirigir unas palabras, ¿sí? Y enseguida el doctor Zanella se va a referir un poco al Congreso de Instrumentación y hará la declaratoria inaugural del mismo. ¿Sí? Gracias.
Y bueno, les comento, estamos nuevamente de beneplácito y hemos hecho todo lo posible por otorgar un evento magnífico, en las mejores condiciones. Esperemos que lo disfruten y enhorabuena, muchas felicidades y sean ustedes bienvenidos en la casa de los mexicanos, en nuestra máxima casa de estudios, en la UNAM Campus Morelia. Muchas gracias.
Bueno, pues muy buenos días a todos, las personas que me acompañan aquí en el presidium, a los organizadores, los participantes del SOMI 34 Congreso de Instrumentación. Y en primer lugar, pues yo quiero agradecer al doctor Israel Betancourt, director del Instituto de Investigaciones en Materiales, pues por haber aceptado acoger la propuesta de trabajar conjuntamente con el Instituto de Ciencias Aplicadas y Tecnología para llevar a cabo el SOMI 34 Congreso de Instrumentación en sus instalaciones aquí en la unidad en Morelia. De igual manera quiero agradecer al comité organizador y al doctor Joel Vargas Ortega, jefe de la unidad, a los doctores Carla Suárez y Rigoberto López y al señor Alan Ortega, así como a los estudiantes y trabajadores participantes en este esfuerzo por su invaluable contribución para llevarlo a cabo. Y esta es ya la 34ª edición del SOMI, Congreso de Instrumentación, que ya cuenta con una gran tradición si tomamos en cuenta que empezó a desarrollarse desde 1980. Y el objetivo central del Congreso pues es, como ustedes lo saben, difundir las actividades de investigación, desarrollo relacionadas con diversos campos de la instrumentación y propiciar el intercambio de conocimientos y experiencias de los participantes. Como ya es tradición, el formato del Congreso combina en 2 días y medio conferencias magistrales, sesiones orales, presentaciones de carteles y actividades socioculturales. Su organización descansa esencialmente en el comité organizador, integrado en esta ocasión tanto por miembros del ICAT como de la unidad Morelia del Instituto de Investigaciones en Materiales, y ha contado con el patrocinio de ambas entidades, así como de la coordinación de la investigación científica de la UNAM y de los programas de posgrado en Ciencia e Ingeniería de Materiales e Ingeniería también de la universidad, y cuenta con el apoyo del personal académico, estudiantes, trabajadores administrativos de ambas entidades. En esta ocasión se recibieron cerca de 120 resúmenes, de los cuales se van a presentar una centena de ellos en forma de cartel o de póster. Y bueno, como estamos un poco atrasados, iba a hablarles un poquito de las conferencias plenarias, pero para ya poder comenzar, nada más les quisiera comentar que como cada año se va a publicar en extenso los trabajos seleccionados, aceptados por el Comité Científico Técnico, y esto formará parte de la publicación electrónica SOMI Congreso de Instrumentación, que se ha venido publicando ya desde hace un buen número de años. Y finalmente, aunque no menos relevante, es el hecho de que este esfuerzo continuado desde hace ya casi 4 décadas se mantiene vigente gracias al interés del ICAT y de algunos de sus académicos, en especial del maestro Gerardo Ruiz Botello, por fomentar la comunicación y el intercambio de experiencias en uno de los campos prioritarios del Instituto de Ciencias Aplicadas y Tecnología, que es la instrumentación, convocando y reuniendo a una parte importante de la comunidad instrumentista de México, esfuerzo que siempre ha sido correspondido por instituciones de educación superior y centros de investigación a lo largo y ancho del país, los cuales generosamente han considerado al Congreso con profundo interés y entrega, como se puede constatar una vez más en esta ocasión. Y no me resta más que agradecer pues el esfuerzo de todos ustedes, su dedicación, y desearles que tengan unas jornadas académicas muy productivas y exitosas. Y bueno, pues pasaríamos ahora a la inauguración. Si quieren nos ponemos un momentito de pie. Y bueno, siendo las 9:37 del 17 de octubre de 2019, declaro formalmente inaugurados los trabajos de este SOMI 34 Congreso de Instrumentación.
Muchas gracias, doctor Sanela. Muchas gracias, y pues vamos a iniciar nuestro programa. Vamos a comenzar con una conferencia magistral. El doctor Gabriel Ascanio nos hará favor de presentar a nuestro invitado, nuestro primer conferencista magistral, que es el doctor Michel Meunier, del Politécnico de Montreal, y dejo el micrófono al doctor Ascanio para la presentación.
Muy buen día a todos. Vamos a dar inicio a la primera conferencia magistral, que va a estar a cargo del profesor Michel Meunier. El profesor Meunier es graduado de la Escuela Politécnica de Montreal en 1978, obtuvo el título de ingeniero físico, y en 1980 el grado de maestro en ingeniería también. Posteriormente, en 1984, obtuvo el doctorado en ciencia de materiales por parte del Instituto Tecnológico de Massachusetts, y en 1985 inició su carrera como profesor en la Escuela Politécnica de Montreal, donde fue promovido posteriormente en 1993 a la carrera al grado de profesor titular. El profesor Meunier ha sido reconocido en diversas ocasiones. Obtuvo el Synergy Award for Innovation, otorgado por el NSERC de Canadá, es el homólogo al CONACYT aquí en México. También es actualmente fellow de la Academia Canadiense de Ingeniería, así como de la Optical Society of America y de la International Society for Optics and Photonics. En 2016, el profesor Meunier fue reconocido con el premio Guy Rocher por sus actividades de excelencia en docencia, y su actividad actualmente de investigación se enfoca al desarrollo de nuevos nanomateriales ópticos, dispositivos nanópticos y tecnología láser para aplicaciones en biomedicina. Ha publicado más de 380 artículos y supervisado más de 120 estudiantes en los niveles de maestría, doctorado y becarios postdoctorales, y actualmente es jefe del Departamento de Ingeniería Física en la Escuela Politécnica de Montreal. Le damos la cordial bienvenida al profesor Meunier.
Thank thank you very much for the invitation, and thank you, Gabriel, especially for inviting me and welcoming me here in Mexico. I really enjoy myself. Oh, I will be on the side of this. Yes. I don't speak Spanish, so you have the choice, English or French. Okay. I did not hear, but I think that I will continue in English. Okay, so all my slides are in English. So the talk will be on, no, just not now. Okay, the talk will be on the use of nanophotonics and ultrafast laser, and for the application in biomedical. I understand that the audience here has a very broad interest, and I will try to be as general as possible and to explain the physics about what's going on. At the same time, then looking at the applications. I'm a physicist. I'm in engineering also, and application in biomedical applications. So my background is mostly nanotechnology and also ultrafast lasers applied to the field of biomedical. Just to tell you first, so now that's the time, okay. Okay, okay, if I click here, it's supposed to change. Now it's not changing. Do I press the good one? Maybe not. Okay, this works. Okay, these two things does not work. Just to make sure that okay, it would be nice if I can change it from here, but this one is to tell you where, maybe to move it up, or if you can use or keep the computer up so that it's just to make sure that does not drop on the floor. Okay. Oh, it was. That does not work. But you have to go back. I have another one. Maybe if it's this one is working. But could you hold this? It's not okay. Hello. Okay. It's just to make sure the computer stays flat. Okay. Okay, let's start again. Okay. So, okay, the idea is to tell you where I come from, and the idea now is that we're located roughly here, and Montreal in Canada is over there, and someone can push the button so that this is a video, so to make sure that the video is in action. It's just that Okay, good, just press. Okay, so if we zoom, and Montreal is an island, so you have Montreal here. At the center of Montreal, there is a small mountain, and on this small mountain, there is University of Montreal. The other side, there is McGill University, and in University of Montreal, there is a Polytechnic. So Polytechnic is this institute here, with other parts here and there. So basically, and this is part of University of Montreal, and University of Montreal is around 60 to 70,000 students. Is quite small compared to UNAM, I understand that. And Polytechnic is world-class engineering with, okay, there is a part that is over there. I'm not sure, maybe we have to push it down, okay? It would be good to do it right now because there will be a lot of things missing from the from that here. Maybe the table is maybe not useful. Okay, it's a world-class engineering. So we're going to have movers now. We're going to move this this table on the side. Okay. It's 8,600 students and 260 professors and close to 50 graduates. It's top 3 in engineering in Canada and the first one in Quebec. Quebec is the French-speaking university in Canada. Usually we say that this is the most important engineering faculty in French for the North America, or for the America completely, let's say. But that's the only one in speaking in French anyway. So the number of students is mostly around 5,000 at the undergrad and around 500 to 700 PhD students in all the fields of engineering. And then this is what you see here, and basically what I'm affiliated with is in the physics and also biomedical. Many professors are double or triple affiliations. So I'm also involved in material engineering. So this is my group. It's a laser processing and Plasmonic laboratory, a picture in May 2019. This is a small part missing here, but it's 2019. So basically what we do is that we develop ultrafast laser processes. And is there any problem if I'm here? I'm closer to you, and at the same time I can read also the text here. We develop and model ultrafast laser, nanomaterials for biomedical, bio-nanoplasmonics, therapy and diagnostics. We have an infrastructure in my lab of the value of around $5 million of equipment, of various plasmonic system, optical setup, nanomaterials fabrication, as well as ultrafast laser. This is a picture of one of the laser here. And as you see, and then we have cell culture facility and biochemistry. I mean, this is something that we do quite heavily, and also we starting to do, and we're doing for some time now, in vivo facility, because doing biomedical is eventually to do in vivo. So this is something that we do in our lab, as well as some other collaborators in the hospital. In my lab, we cover fundamental and physics aspect up to the application in medicine. I try to do the complete translation. This is not easy. This is very difficult to do also, because as a specialist in the physics and engineering, sometimes you don't know too much about, the other side here. So you need good collaborators. So from the physics point of view, Plasmonics, I will explain that. These are nanomaterials that we are producing. Imaging, physics of ultrafast laser in relation with plasmonic nanostructure. I'm going to explain to you some of the aspect to that. On the engineering side, it's to apply this here to the development of either devices or systems, laser nanosurgery system. This is what I'm going to cover in a few minutes, and also develop the drug delivery, and also in application in medicine. As you're going to show, we're developing application in the field of ophthalmology and also pathology and many other topics that I'm not going to cover. Okay, in this talk I'm going to cover two topics. The first one, this is the title here. The first one is to perform nanosurgery, and I'd like to cover that in a very general way so that you have some good understanding about what we are developing, and also bioimaging. This one will take maybe 2/3 of the talk, and the other one the last 10 minutes or something like that, just to tell you that the first one is that they elaborate much more. So let's start on this one here, and I'm going to start very far in terms of history, and I like history. So usually I covered this part here, the scalpel in the history of the medicine. Here in surgery, this is a book written in French. I'm not sure if it has been translated in other languages. But the history of surgery started, tens of thousands of years ago. And of course, in the Middle Age, these were the instruments, and of course, the scalpel of the 20th century this is what we are using right now. There is a limitation when you use a real scalpel, like a knife. This precision is limited to the blade angle here. And of course, you can use a laser, and that's why I'm here. You can use a laser. You can use a laser to perform very fine surgery, and this is being used in many types of approaches of using laser. That's mostly for the eye, but some other aspect in surgery that this is done for, of course, for surgery, but also for imaging and sensing and so on. If I try to focus a laser in the biological media, from a very basic physics of focusing a laser, you focus a laser of the order of the wavelength, roughly. In the visible, it's around 500-600 nanometer. The problem is when you absorb it, it diffuses the energy around, and this dissipated energy will go over many wavelength. So it's very difficult to do some kind of what we call a nanosurgery. It will be something much more like a microsurgery or even larger. The question is, can we do better? And of course we can do better, otherwise I would not be here. The idea is to use different types of laser. In this book that I mentioned a few minutes ago, the great surgeon of the future will be a nanosurgeon. It's just to perform surgery on a nanoscale. Perform surgery not in tissue, of course, but also directly in the cell, and maybe in the very fine scale in the cell. And that's the idea that we would like to develop. Ultrafast laser can be used for that. An ultrafast laser has a pulse width in the femtosecond time frame. Okay, so this is very, very short. Just to make an idea, if you look at vibration in molecules or vibration in whatever material, this is mostly like in the picosecond and even longer. So we're 1,000 times lower. And this allows you to perform different types of surgery or ablation on a very limited region. It limits the energy diffusion, it limits the heat affected zone, and also due to nonlinear phenomena that I'm going to explain in a minute, you're going to have something that is very, very fine. So this is an ultrafast laser. For those who have never seen that, it's not a laser that I'm standing in my hand. This is a solid state laser in the visible. I mean, you can buy that for, I mean, few dollars or probably $10 or something like that. Okay, a laser like this is, I have two. I mean, this is the smallest one for me. I have another one that is bigger. Basically, I mean, a laser like this costs maybe $300,000 or $400,000 or even more. It is a laser, but basically it is a lot of optics inside and materials, and fine-tuning that is very particular. So basically, usually to use an ultrafast laser, it requires someone with great skills in engineering and in optics, and usually, I mean, PhD students actually. This is a picture that was taken by a colleague of mine a long time ago, almost 25 years, 1996, almost 25 years. It shows that when I focus a nanosecond laser, this is more like a conventional laser. This is stainless steel, it's nothing to do with biomedical, but this is one of the first pictures that shows that basically when you do this, it doesn't look like there is something that is very nice, but with a femtosecond laser, it's much nicer. And as you see, the region around here is very limited. So in that case, I can do a much better ablation, much better even surgery with that gives rise to a heat affected zone that is very limited. So this is a very interesting approach. This has been developed for, 25 years. So basically, with a nanosecond laser, a little more like a physics aspect a little bit here, going from one level to another level with an 800 nanometer. 800 nanometer has an energy of around 1 electron volts or a little bit higher. And basically, if one wants to get from a level to another level here, with a nanosecond laser, that does not work. However, with a femtosecond laser, there are so many photons at the same time that one can get many steps and arriving to the top level where you start having the ablation, dissociation of molecules, and so on. So this multiphoton absorption, because of high density, is the source of the possibility of doing nano surgery. And this has been done for many decades, actually, since the beginning. So if I focus, this is a beam that is focused on the plane here. And if I look at the intensity on this line here, it's like a Gaussian here, and the intensity is high, and the treatment would be function of high here. However, if it is multiphoton, if I need 2 photons, the intensity would be square, and the treatment zone will also be square, and so on, and then by going at a much higher level, you get something that is very small. So this idea has been developed for many years now, and some of my other colleagues, Eric Mazur, that I know he is at Harvard, can do cuts at many hundreds of nanometer, or even, a cut like this, with 100 nanometer. So limitation in the tens of the hundreds of nanometer, based on nonlinear effects. Can we do better? Can we go over that? Can we go over the optics? Can we go, and that's the way of using nanotechnology now. So now what I'm going to tell you is a combination of using an ultrafast laser, as well as nanotechnology. I'm going to use these two approaches. These two approaches are like in the extreme. That the one is ultrafast, is femtoseconds, so very short time, and nanotechnology, nanometer. And and we can do better. And even that, we can do more. And that's the idea of this. We started on that 10 years ago, and basically it was like a lab curiosity. And right now, in biomedical, we are doing that to solve illnesses in ophthalmology. That's one application that I'm doing. It's just to push as much as you can the lab curiosity, because it was a lab curiosity at the beginning, to the application. And that's one of the messages that I can give to some of the students here. Push your idea at most. Sometimes you can get something pretty interesting. This is what I do with my students, actually. Okay, let's come back to, I'm going to use plasmonic nanoparticles. So, nanoparticles. So I brought some nanoparticles here. So I have it's very small because yes, I'm going to just put it there here. Okay, at the break, and then you can see that. Okay, these are gold nanoparticles. And then the first time that you see that, so sometimes you say, oh, the gold nanoparticle doesn't look like gold when I look at that. It's red. Yes, this is 12, 14, 15 nanometer. Actually, in this, gold is a metal. In this metal, there is a lot of electrons, and then a little bit of physics now about what is going on. It's called a plasmon. There's a collective free electron oscillation in the metal. So basically, what is happening is that all the quantity of electrons in the material is just oscillating upon the irradiation. And by developing this, it happens that you have a peak at around 5, this is the wavelength, so around 570, something like that, or 550 nanometer. So this is in the green. So you should say, there's a peak there in the green, but what I look, it's red. It's red because this absorption peak and scattering peak is all in the visible. So this is the central, the visible. Here you start to have the region where it's the red. So basically when I shine the light through this, everything here is absorbed or scattered, and what goes through is red. So that's why it's red. It's red because of this. It's red because of this plasmonic peak. So this idea now that I can use as a phenomenon. I mean, it's interesting to see and to study and to use, and actually we're using that in biomedical. So the idea now that what we have developed is the following. When you look at the phenomena here of an irradiation, there is a near-field amplification close to the particle. I think there will be a talk tomorrow, I think, on this near-field and source phenomena. So basically, this nanomaterial, when it is irradiated, let's say at 800 nanometer, polarization that is linear, you have a near field that is much higher, around mostly 5 or something like that, close to the particle. And this is, I will be coming back to that in the next minutes. So basically, what I have here is much higher field that is amplified close to the particle. Actually, if you remember your basic physics, the laser, the irradiation, the energy into an irradiation is E squared. So basically, you have here 25 more times energy than here. So it concentrates the energy. So basically, what it is, it's a lens. It's a nano lens that is concentrating the energy close to the particle, And this can be used actually, and this can be used for developing that. And that's the idea that we did, long 10 years ago, by putting nanomaterials on a piece of silicon, and upon the irradiation of a femtosecond laser, we were producing nanohole. So I brought here, let's pretend this is a nanoparticle, okay? Okay, and so this is a surface. I irradiate the surface on that scale. Pretend that this is, 100 nanometer, or let's say 50 nanometers. Okay, and you have a big surface here. And then when you shine the irradiation, the fluence of the energy into the pulse might not be sufficient to do something here or to do something everywhere. But close to the particle here, there is this field amplification. And the field amplification close to the particle can give rise to this phenomena here of producing a nanoablation. So this is due to the field amplification. It's because this nanoparticle is acting as concentrating the energy at a specific point. So that's the idea. This is at the beginning like a lab curiosity, that we were putting nanoparticles and just looking with the ultrafast laser and say, why? We're doing little hole, and what can be used with that? And so I come from the field of microelectronics, optoelectronics, and this can do maybe some kind of nanoablation. The idea is that, can we switch to another material? Let's switch to biomedical. I'm not a biologist, and at that time I didn't know too much about biology. So the idea is that let's push the idea quite further. Okay, so I try to convince biologists to say, I'm going to put nanoparticles and irradiate that. Okay, so what can I do with that? So what can I do with that is making little hole here close to the cell. So I can perform a surgery with the same basic principle. The principle will be that close to the particle, I will create something, and that something, which is like, let's say a nanobubble or something like that, that will maybe perforate the membrane, that will do something on a nano scale. And that's the idea that we started almost 10 years ago of pushing that lab curiosity to the point of maybe doing something that can be useful eventually. So this is what I call a multi-nano scalpel. So basically, we're using an ultrafast laser. The wavelength of this laser is 800 nanometer. 800 nanometer, as you see here, the absorbance in biological media, whether it's a melanin or water, there is a region where the absorbance is lower. Actually, if I use I don't know if you see the red through my finger here. Basically, this is in the red, but in the near-infrared at 800 nanometer, it penetrates centimeters. So basically, that's the idea of using this type of laser. Now, the idea, if this is the cell here, this is the big cell, and this is one nanoparticle that is there, at 800 nanometer, I will not affect the cell. But around the nanoparticle, it will concentrate the energy and start doing something. And that's the something that I want to use. Okay, so if I use a conventional laser, I focus it, and I can do a, let's say, mostly micro scalpel. If I put one nanoparticle, the same thing that I'm put on here, I will do something that is much smaller, and that's the idea of that. But let's do something else. Let's do something further. What I can do with a nanoparticle here is that I can attach some biomolecules that will recognize something on the cell. And this is what biochemists and biologists are doing for some years now. as a physicist, sometimes it looks like recipes that we have to develop, and this is the case. In my group, I have specialists in that. So basically, if I can have on the nanoparticles some biomolecule that will recognize specifically some sites, and then I do the irradiation, I will do a surgery at a specific place, depending on this biochemistry, depending on this approach. It's like a seeking device, okay? A seeking device that will seek, and then it will be at a specific place, and then I do an irradiation. And so that's the idea. It's a multi-nanoscalpel, but at targeted place. So if I want to do it at this place and not the other ones, and I can functionalize, we say it's functionalize these nanoparticles with some specific molecules that will recognize this, and then I just do an irradiation. This is what I want to do. So basically what I'm going to do is put these nanoparticles there and then do the irradiation. I can do this multi-nanoscalpel all at once. So I'm not doing just one nanosurgery, but I do a nanosurgery at many places all at once. Actually, one pulse, and I'm doing something. So, what can this be used? Okay, let's do first. Let's go a little bit of the physics of that. And as a physicist, I found this process very interesting. It is in the femto. It is in the limit of what you can do in terms of in terms of the extreme that they explain. It's femtosecond time frame and nanometer scale. And there's a lot of things that is happening on this scale, and it is multidimensional. And then this is expanding in time, and then expanding also in space. So what can happen? Okay, upon the irradiation, I can heat up the particle. I can heat up what is surrounding the particle, and a biological media is mostly water. So we are heating up water actually. We can produce a lot of electrons. It's like a plasma here. It's like a plasma, but the plasma can be very high in energy, very high in temperature. It can reach, according to our calculation, tens of thousands of degrees, but during 100 femtoseconds, and in the space of tens of nanometers. So we're not killing cell with that. It's just that this is dissipating out actually in the cell after that. So after that, it releases the energy to the water. There is a relaxation that can give rise to some kind of pressure, because if you increase the water during a very short period of time, you will produce some kind of pressure that is coming out of the particle. And this is something that can be used, and eventually this will give rise to a bubble here. So it's acting like a nanolens, and as a picture, if you did not understand all the physics about what is going on, it's like a nano heater. It's like a system that produces a nanoplasma during a short period of time. It's like a nano tsunami, like a tsunami in water, coming from the particle and going away, and then producing nano bubbles. Okay, we published that some years ago. Okay, yeah, we did a lot of calculation and modeling and so on. I'm going to skip that. Just to show you that according to all our modeling and so on, and this is multi-physics, we can model and understand the amplification generation here of this nanoplasma that can last, I mean, tens of picoseconds. A pressure wave that starts in the picosecond up to nanosecond, and this bubble that starts at this value. This is the results of all the calculation, but I'm going to skip all that. Okay, I'm talking about bubble. Bubble, it's real. We have measured that. There are techniques that we can use. This is the little video that we have here, and basically we can pump and then with a probe and look at this bubble in time. And as you see here, I mean, there is a bubble that is increasing and then decreasing on the scale of, let's say, 15 nanoseconds. So we're producing a nanobubble during 15 nanoseconds, but close to a cell. This will perturb the cell up to the point that it will be perturbed during many seconds, even that. Why the particles are not broken? If you have a surgeon that is using his scalpel for surgery, you don't want to have the scalpel to break, and this is very important. So here we're using the laser. This nanoparticle, we don't want that it breaks. The idea is the fact that there is low absorption at 800. This is the curve of absorption, and as you see here at 800, the absorption is very small here. So there is not a lot of energy that is in the nanoparticle, but actually where this energy is this amplification. It's outside the particle. That leads to a generation of plasma and cavitation. And this plasma and cavitation, that is the source of what we can do now as a nanosurgery tool for performing all the steps. So if you did not understand all the detail of this, which I went very fast, I agree on that. It's basically this nanoparticle is actually acting as a nanolens. It's concentrating the energy, but in a lens we concentrate the energy here. And with the nanoparticle, it's like we're concentrating the energy on each side of the particle. So it's a different ways of concentrating the energy, but this is some sort of a lensing effect. What can we do with that, with the various nanoparticles, process? It can be a nano heater. If I tune it to the way that it is producing heat, it will be a heater. This can destroy cancer cell, for instance. It can produce extra charges that can break specific chemical bonds. It can produce a pressure wave. It can produce some sort of a mechanical pressure that is expanding close to the particle. Just to remember, we are on a nanoscale here, so this will be a pressure on a nanoscale. And then, of course, bubbles. I'm going to show you an application for a bubble, and this is called optoporation and transfection. So the word opto, you see what is opto, it's the light, and poration is that it will gives this cell to become with porous. Okay, that's the idea behind that. So that's one of the application of this tool. So basically, I don't know if there are biologists in the room. Probably every time I do that, I mean, there's nobody. Yes, but maybe there's no biologist. Okay, I'm not biologist neither. So this is the way to describe what I understand from what it's called transfection, or it's to include, it's to go over the cell membrane and to include some biological species into the cell. So a cell is formed by a nucleus, and the membrane of the cell, essentially, we have to go through this membrane. And going through the membrane, this is one of the difficulties. So one application here is to use the technique for doing that here. So this is conventional way by biologists. Biologists are using quite a lot. You know what this is? That's a virus. So they use viruses. So they let's say HIV or something like that. So they empty the content of the virus and they include what they want. And then these viruses, they are very good on just injecting in cells something. Okay, so this is very popular. So some people would like to use something that would not involve viruses. This is more physical approach. An electrical shock. You have your cell, you put electrodes, and you have an impulse, and with that impulse, you create pores on the cell. Most of the cells die because it's an electrical shock. Laser radiation, this was also done, and my technique is something that is pretty new on that. So the idea is transfection is to include foreign DNA or something like that into the cell. We need to perforate the membrane. So this is what we are using here. So we put nanoparticles on the cells here. This is not a snowstorm, as I usually see in Quebec. This is, I mean, an imagination of them in gold nanoparticle going on cells here, and then as they go on the cells, and then one produces one shot, some kind of bubble or perforation, and then this membrane is perturbed during sufficient time so that you can have something that is from outside going inside. So that's basically that. So this was shown in vitro. So we have cancer cell line here. This is a cancer cell line that we can grow on the in vitro, and then we just scan it here, and we don't scan here, and then we develop everything and so on, and I'm skipping all the details, and basically when it's green, it's because we are successful. So this is working quite nicely actually, and in a much better way than some other techniques that biologists are using. And I'm going to skip that because there's too much detail in terms of that. Okay. I use the same technique to stimulate neurons. Neurons from the brain here. Neurons from the brain. The idea is to stimulate neurons so that there is an action potential between the various neurons. And so this was published some years ago here. Basically, the idea here is that with neurons that we can put nanoparticles, even nanoparticles of different types, because here I'm talking about gold nanoparticles, but we can have different types of nanoparticles. So different types of nanoparticle that will absorb or make different types of effect. And the idea would be here to stimulate with a near-infrared laser by 800 nanometer. Cells are weakly absorbing in the near-infrared here, and then stimulate the neurons so that there is some kind of effects going in different directions. So we can play with that circuit actually with the light, and this is on a nanometer scale in terms of stimulation. So with that, I can possibly heat up some neurons or producing a mechanical wave on it so that I can interact with some neurons and may even produce nanobubble. So we have done that, and when it is becoming more green, it's because basically there is an action potential that is done. So this is the way now. This is a tool for neuroscientists that can use that and stimulate neurons at different places. So that's one application. We're moving now with a technique to use that in ophthalmology. Since the beginning, I was trying, remember, it's a lab curiosity, putting nanoparticles on a piece of silicon, irradiate and making little hole. Now I was thinking at the beginning, how can I use that in vivo? How can I use that to treat illnesses? And the best way to use that is to use a laser in the eye. You would say, never use a laser in the eye. I mean, this is not something that I should do, but ophthalmologists are doing that all the time. I mean, they use that, and you do, surgery, and you mean eye correction. Maybe you have some people that has done eye corrections here in the room. Nobody. Usually, one. Yes. Just so usually it's getting more and more popular. Okay. So the idea is to use that, and ophthalmologists are using quite that heavily. Here, it's the idea is to cure now, not to do some kind of surgery or reconstruct the cornea, but is to cure. Cure means that I can deliver genes, or I can deliver drugs directly in the cell in vivo. And that's the idea here that we are pushing. Ocular diseases affect millions of individuals worldwide. These diseases are usually either incurable or only transiently improved with current therapy. And what we are producing here is to use high-throughput non-viral gene delivery treatment, is to use this technique now that it does not involve viruses. It involves a combination of laser and nanoparticles. We inject nanoparticles into the eye. Yes, this is something that is done currently by ophthalmologists. They inject something into the eye so that you have a specific, drugs or something like that. So this can be done. We're delivering small interfering RNA into retina cell. This is something that we are incorporating so that it will silence, let's say, some illnesses in the cells of the, let's say here, the retina. We try to address the age-related macular degenerescence. As you're getting old, sometimes what you see is black spot when you look. You only see on the periphery, and the center it's black. This is the age macular degeneration that is getting more and more popular because population are getting older and older. So we use that on animal. This is our little friend now. And we do two double targeting. We functionalize nanoparticles and we use the local laser irradiation directly in the retina. Okay, so you need students now that can handle a small animal and then use with the small animal and to irradiate the laser into the eye. There is a lot of ethics to be done and so on, and it's all in agreement with all that. Okay, this is to show that the particles are at the good place. Not going too much detail on that. This is conventional systems for that here, and this is the result. Basically, when you see it red, so we do that in vivo, and then the animal is sacrificed, and then you take the retina, and then you look under fluorescence spectroscopy, and what you look here is the red. The red is because we put a red fluorescence close to the sRNA, and basically this is in the cell. So depending on the energy and so on, we show that it is working. Now it's a question to optimize it. But the first thing I did before writing a patent, because I have a patent on that, because this can lead to a new way to treat illnesses into the eye. So basically here, what we look, and not going into much detail here, is to say that either, the injection or with the irradiation, it's non-toxic. 1 week after, the number of, the growing cells and everything is okay. So for me, this was the last test to write the patent, and now we have a big program on this, and this is what we are developing. So this is a conclusion of my first part. I'm not sure how I'm going from the time, but this is only my first part. The second part is much shorter, but we are developing this, and we have a big program now financed by the government and at different levels, and that we are developing that right now with on the retina, as well as we're just starting on the cornea, as I was going to mention the last page. So actually, laser is mostly used to cut, and basically, I hope that one day that we use that for therapy as well. Okay, so we have many papers on that, and if they are interesting PhD students, I have already one from UNAM. There are other ones. They are pretty good, so I'm going to come back to that. But basically, these are some of the papers on this topic. Okay, in the last 10 minutes or so, or maybe 10 or 15, I'm not sure how I'm going in terms of timing. We're late anyway, so this is what Gabriel told me. This is Mexican time, so a little bit late. So what we have here are gold nanoparticles, and I brought here silver nanoparticle. Depending on the dimension, depending on it's a different colors. And the idea is that there is some space between the two here. And this is what we have developed over the years. So we have a patent on this that is accepted now. When you have a patent, you go through provisional patent, and then you have to go through all the various other steps and discuss with the patent agent, and it lasts forever. But basically, we have this. So we have all the different colors, and basically what we can do is fabricate any type of any composition of any size, and the geometry is a sphere. So these are nanoparticles of let's see here, 56 nanometer with minus 10%. So this is what we need, and the idea here is to be able to, there are, different dimensions at different colors, that it is seen here. Almost multiplexing for at least for markers. So we can functionalize, and this is biochemistry, and I'm not going to go through that. So the idea is the following here. So we have many nanoparticles, different colors. So the idea is to look at these different colors. So you have different colors. You put some kind of biomolecule on it so that it will target specific cells, and then from that you look at the optical system, and then you can have, this is one cell with different types of nanoparticles of different colors in it. This is what it's called the hyperspectral imaging. So this is an optical setup so that you can have X, Y, Z, and lambda. So it gives you a lot of data. So a lot of fun for those who would like to do some image analysis and so on. So that's the idea, is that when you have these nanoparticles, by irradiating, this will give you different colors here. And compared to other approaches, it is very interesting and very good. There's no photobleaching, where, most of the fluorophores are photobleached. Photobleach means that when you irradiate, the intensity decreases. Here, this will last forever. These nanoparticles were fabricated, many years ago, and the property is always the same. So this is one system here. A lot of detail on this page, but basically these are cancer cell line, breast cancer cell line, and then you have the nanoparticles over the breast cancer cell line, and then you can do you can see the various nanoparticles. This is another system here. This is to reconstruct from an hyperspectral approach, that you can reconstruct and identify each nanoparticles and then reconstruct and make. This is a false color. This is not the real color, but this is a false color to tell you where they are and so on. So this is another way of doing this type of analysis. This is multiplexing. So we have a spin-off company, VegaPhoton, that is essentially developing these type of devices, and based on various nanoparticles and various systems. These systems can be integrated in many types of optical setup that we have, and so we are developing that here. And this system here is something that we are developing. For those of you who know Jennifer, this is the end of Jennifer. I'll come back to her at the end. Okay, so that's the way of looking at different colors depending on the irradiation by the LED from the side. This is what we see. And so the idea is to use that now to help the pathologists to make a good diagnostics. So this is a this is by a conventional dark field system where there are nanoparticles, but you barely see them, but just by switching to our illumination. So it's a question of illuminating from the side, or instead of from the top, or from the top or the bottom. what you're going to see is that, let's say here, you don't see nanoparticles. Maybe with the light, it's not that great, but here, what you see are the nanoparticles here. So this is not a galaxy with stars. There are cancer cells with nanoparticles on it. And so this is the things that we are looking, and that we are doing image analysis now on that. One of my students is doing artificial intelligence analysis on that. So basically, as I told you, with that, with hyperspectral, you have X, Y, and Z, so you can have three-dimensional, plus at each pixel, you have a lambda. So you have a four dimension actually to treat. Okay, so here this is a scan of a cluster of a cancer cell that is actually occurring into the eye of children. And so this is not in the eye of children. This is a cancer that we grow on a phantom here. And we use our side illumination. So the little video that is, I think it's stopped now, but basically that what we see is that there are particles everywhere, so that we can now analyze the various position and then do some kind of identification and also some treatment. The idea here was to irradiate and kill the cancer. And this is what we have done. Basically, if you irradiate this with a lot of nanoparticles in it, you destroy everything. And so this is the way that we are looking at destroying cancer cells aggregates into the eye of children. So this is identification of 4 types. Here, there are 4 types of nanoparticles. It is not that clear here, but so we use our system, and then from that do the analysis, and this is the position of each of the nanoparticles into this plane, and then we have various planes that we are looking at. So we have plenty of papers specifically on that. So my time is almost out, I think. So I would like to conclude that we cover 2 types of biomedical applications using nanophotonics and ultrafast laser. One is using nanoparticles that can do 3-dimensional multiplexing imaging. So a lot of fun for people that are developing hardware optics, also developing nanoparticles, and also image analysis using various approaches, and then use the technique with the ultrafast laser to do various types of process and technology for the application in the medicine. I'm looking for a PhD student. Right now, we are financed quite well. The problem is the people. I need people, so this is part of my group here. Picture taken in May. So there is place for many others students on this type of project. And maybe you have recognized one person. For some of you who were at UNAM in Mexico, so this is Jennifer Zapata Farfan. Farfan, how do you say? Farfan, yeah. I usually call her just Jennifer, okay? So she's doing pretty well after one year. She's PhD candidate in biomedical. That's the laser that she's using now. This is an ultrafast laser amplify. This is a pretty nice tool. And this system is with 6 tables all connected together. Her project is to translate what we are doing to cornea. So we're using cornea from a mouse, and then there are biologists on the project. There are also some other expert in biology. She's the expert on the irradiation laser and looking at the final result. Okay, so that in my group looking for a PhD student, postdoc, mostly student interested in laser, in more theoretical aspect, more applied aspect, and nanoplasmonics, as well as in for biomedical, and all the spread from the basic physics to engineering to the application in medicine. So you can send me a message or talk to me. I would be here, I mean today, I think. Not sure tomorrow, but today. If I put my hat now as a chair of the engineering physics now, I'm the director of engineering physics, we will be looking for several tenure track opening that in the Department of Engineering Physics is mostly involved with optics and nanotechnology and quantum technology, application in biomedical, energy, and so on. And so this is not, this is in coming years, in the next 4 years, we'll be hiring many new professors. The standard is quite high. We are really looking for a potential candidate that have a good CV in research. Okay, I think I will stop here. Okay, that's the end. Okay, so I would like to thank you of your attention, and I'm open for discussion, comments, or anything. Okay, thank you.
Well, thank you very much, Michel, for this very interesting talk. And we have some minutes for questions.
Hello, and thank you for the presentation. It was really interesting, and I really like what you are doing. My name is Andres. I work on image processing. I'm a master's degree student, so I'm actually looking for a PhD position. But I would like to know about that aspect of your research. How do you use image processing, and how would you like to use it from? What work is to be done next?
Because of the noise, I'm not sure if I understood correctly. So you are interested in image processing and so on. Right now, we're just at the beginning of trying to see how we can handle all these, all the data that we're producing with our optical setup. So we are at the beginning of that. So I have one PhD doing this. He is much more on the let's say computer engineering, handling a lot of data. So his background in physics is okay, and I have another student that his background in physics is very good in developing the system, but less on the other aspects. So it's complementary team. I think it's interesting. So the idea would be to develop the combination of hardware and software, so that we can have at the end something that the pathologist would just look at it, push the button, and you have the result. The pathologists, the way that they do it, the pathologists that I'm discussing with, they are, I mean, pathologists, I mean, most of the time, they do their job at the same time that they teach. They look under the microscope, and they have this cancer tissue, and they make a decision. If they look on the camera, they don't like that. They don't want to have an intermediate. The digital pathology is not something that is well developed. So this is one way. There are many other ways. So this is the development of pathology right now, is to try to get these images, but to translate that somehow that it will be understandable for them, And this is a very big step for them. And actually, most of the pathologists, they don't want to look at this at the screen, and even less at the screen that has been processed. You image your process for different things, yeah, they don't want to look at that. What they want is to look specifically at the directly at the image without any. Anyway, so there is a lot of job to be done on that. This is what I wanted to say. Okay, yours, you have yeah. Okay, I will bring you the microphone.
Thank you very much. What about the inflammation of the cells? I mean, inflammation when you hit, for example, in the knee or in the here or here, your tissues get inflamed, and so the cells are inflammation. I don't know if the right word. But I mean, you are using the nanoparticles and all, and the photon laser for ophthalmology. But what about the muscles, the tissues, for example, for the knees?
Maybe I can say we can just apply this to other types of other types of medical problems, and I'm open to that.
No.
The idea is to talk together with some medical doctor that will say, okay, I'm having a specific problems for that, so maybe my technique can be a solution. And but I'm not a medical doctor. I'm the inventor of the technique. Now I'm looking for application. So I have one application in ophthalmology, and that really takes me a lot of my time. But other application will be very, very interesting. So this is something that we are working in parallel, but of different types of it. Let's say like if you have cells from your body, you have it in vitro, you can do your treatment, and then you re-inject the cell. And then for treatment in cancer and so on, this is very interesting. The problem is that it requires, a large quantity of cells, which is sometimes a little bit more difficult with my technique. But it's a question of scaling up. But this is something that can be feasible. Yeah. Is that microphone working correctly? Or? I need to hear her, so I will come closer. That would be better.
Are you sure that the nanoparticles is exactly when you like to put this?
Okay, so the question is, I'm putting the nanoparticle, are you sure that you're putting the nanoparticles at the good place? And so the question is, this is a very interesting and important question, because if we think the particles at one place and they are not, the process does not work. So the idea is to inject it, let's say for the eye, is to inject it at a specific place. So ophthalmologists are good for that. And I have one PhD student that is getting pretty good of injecting into the eye of an animal at a good place, and then try to see if the particles are really there. And so yes, they are there. So they are at the good place that we want. for the retina, and for the cornea, last result on the last weeks, yes, they are at a good place. And then you shine the laser at the place that you want. So combination of particles and laser give you the process. Yeah, of course, in the eye, I mean, it's a viscous material, so we're facing a problem of fluid dynamics, basically. We have to inject at a specific place and make sure that particles are going at the good place. What type of? Yeah.
Laser nanosurgery. What kind of eye diseases can be treated with this technology?
The eye diseases, as I mentioned, is for the retina is the age macular degeneration. This is what we're looking at. For the cornea, a lot of people are doing, a transplant of cornea. But there is some kind of disease that I have to remember. My colleague she's a ophthalmologist, surgeon for cornea. There is a specific illness that I have to remember. Maybe I should remember the name because I'm talking about that. So basically, this illness is it cannot be cured. We need to do gene therapy. So this field is called gene therapy, which is a very interesting approach for illnesses, and it's a long way. It's a very long journey because just showing on one animal and then another one and then looking at the recovery, it takes years and years and years for doing that. This is only on animal, and then you have to go through phase 1, 2, and 3 for human, and usually it's the phase 1, 2, and 3 is the number of 1 million, 10 million, $100 million. We're not there. Right now, we have a little bit less than $1 million to develop within ophthalmology, an animal model. Yeah. Okay.
Okay, in the name of the organizing committee, I would like to thank you for this talk. And I have two gifts for you. One of them is this diploma.
A diploma?
Yeah. Another one. And the second one is handcraft made in a place very close to Morelia, Sparacho, in which they produce guitars.
Okay.
And this is also for you.
Can you play guitars? This is not a nano guitar. It's still a micro guitar. This is yeah, yes, okay. But thank you, Gabriel, and thank you for your attention. Very interesting.
Bueno, continuamos con el programa. Estamos, como ustedes ven, bastante retrasados. Aquí ya se establece la sala 1 para las presentaciones orales. Las salas 2 y 3 están fuera del auditorio, enfrente de la barra, hacia el otro lado. Por favor, quienes participan allá o quieren permanecer aquí, se pueden mover. ¿Sí? Bueno, ahorita